it does. So it's just basically about DNA and its structure, how it condenses, its interaction with proteins. So it's more of a structural lecture on DNA and the sort of things the DNA will do when it has to condense to fit into the chromosome structure in the nucleus, because we're dealing with very long molecules in general, apart from extra chromosomal DNA, which are called plasmids, which you generally get in bacteria, which are generally a lot shorter. And we have issues like super twist, super coiling to pack the DNA, again following the laws of thermodynamics. So energy is very important. As a consequence of that, certain things happen or can happen to the DNA molecule, which are very interesting and which we'll explore, and which are also explored in question 2. And the deadline is not that long away, is it, for the assessed questions for this module? Is it end of the month? 3rd December. Okay, so still a while to go. Okay, so DNA makes proteins, and the so-called central dogma of nucleic acid and protein synthesis is that DNA is replicated. It's the basis of life, that DNA replicates itself. Obviously, it's got to have a supply of nucleic acids and carbohydrate and phosphate to be able to do that, but we assume that those substances are in place and DNA replicates. And then it's transcribed to RNA, which takes the message from the specific genes or coded in the DNA molecule, which require translation into protein. So when the correct signals for producing the protein are there, then the DNA is transcribed to give RNA, and the RNA, the messenger RNA, takes the signal outside the nucleus to structures called ribosomes, in which translation of the genetic code takes place to produce the correct proteins that are required. So it doesn't make any protein, because that's just a waste of energy. Proteins which are specific for particular functions, enzymes or structural proteins or whatever. And then you can get post-translational modification of the protein, such as glycosylation, then becomes important in molecular recognition phenomena. But this is the so-called central dogma of nucleic acid and protein synthesis. It's a nice diagram from Sverdrup, and it's a very clever mechanism, really, quite brilliant. So this is the message, which the messenger RNA is taken from the relevant bits of the DNA with this code, and these wonderful molecules called transfer RNA, again, given the right signal, will basically bring the correct amino acid corresponding to the code on the messenger RNA. So they bring the amino acids in sequence, and then the peptide bond is formed. And then when the stop codon, this 3-letter code, or these 3-letter codes here, are approached, then the synthesis stops. And this gives proteins the precise molecular weight monodisperse structures because of this template, which doesn't occur when you come to sort of polysaccharide synthesis. It doesn't have this superb template. And all this takes place in these multi-protein structures called ribosomes. And those proteins themselves in ribosomes have been produced by a very similar, by the same genetic translation process. So it's a very, very clever way of the nature's derived of producing proteins. Right, here's a bit of history behind DNA. DNA was first suggested, DNA was discovered well before this date, 1946, but it was 1946 that Avery came up with the proposal that DNA was responsible for the genetic code. Before, it wasn't quite clear which molecules were involved, but by 1946 Avery came up with the proposal that it was the DNA which is the main constituent of genes. So with the film, the Watson-Crick film, where Watson and Crick meet each other, and what are the most exciting things in science today or back in 1950s, and it was the genes. And Crick says, we think it's something to do with the nucleic acids and things. So even by the 1950s, there was still some doubt whether nucleic acids were involved with genes. Then around the same time, that's when the hydrogen bonds were discovered between the bases of the DNA. It was already known that these were long sort of linear types of molecules, but it was 1946-47 that hydrogen bonds were discovered here in Nottingham by Creeth, Gulland and Jordan. 1950, the first X-ray pictures of DNA, Rosalind Franklin, but also a group at Leeds University around the same time produced pictures which could be interpreted as evidence of a helix. It's just buried in someone's PhD thesis, and only later was it discovered. A bit like the hydrogen bond story and the structure, the Creeth structure for DNA. Then another breakthrough came in 52 with the Shawgraph rules, with the equivalence of the C and G and T and A bases, so that the numbers of cytosines match the guanines and thymines and the adenines. They were the so-called Shawgraph rules, which proved crucial for the final evaluation of the DNA double helical structure by Watson and Crick in 1953. Since then, the messenger RNA was discovered in 1961 by Jacob Monod and also Brenner and Messelson. And then in 1973 was the X-ray structure of transfer RNA, showing the classical cloverleaf structure. So these, because many more things should be included here, but these were probably the main events in terms of the structure of nucleic acids. And the primary structure of DNA and also RNA follows a similar sort of pattern. You have this phosphate sugar backbone. These are sugar residues. In DNA, it's deoxyribose. In RNA, it's ribose, RNA, DNA, and linked quite different from the glycosidic bonds, which Tim Foster we talked about last week, but by these phosphodiester bonds. So the phosphate residues link the sugar molecules together. And unlike proteins which have just two conformational angles around the peptide bond, the DNA, RNA have five conformational angles, four of them associated with the phosphate. The other one here between this carbon atom here and carbon atom number 5 here. And this gives the molecule much greater conformational freedom. So they tend to be linear types of molecules. In the case of RNA, very flexible. DNA, the flexibility is compromised because the formation of the double helix. Then you have these bases, C, T, A, and G. In RNA, the thymine is replaced by a uracil group, which is a related base. So the nucleotides are the bases: adenine, cytosine, guanine, thymine, and uracil in RNA. And we call the combination of the base with the sugar as nucleosides. So adenosine, cytidine, guanosine, thymidine, and uridine. So nucleosides, there's nucleotides and nucleosides. And the bases themselves fall into two groups. The purines, guanine and adenine, have these two ring structures, and the pyrimidines, cytosine, thymine, and in RNA uracil, have these single benzoyl ring structures. So according to the Shawgraph rules, C binds to G and T binds to A via these hydrogen bonds, and the difference between C and G and T and A is that the CG is stabilized by 3 hydrogen bonds, so it's a stronger bonding, whereas the TA link is by 2 hydrogen bonds only. It was thought that there was a bonding here, but the distance between the 2 residues is too far to promote this. You can get post-replication processing with the addition of methyl groups, and also post-transcriptional processing in a similar way, modifying the bases. And DNA is kind of effect of opening out the double helix, exposing bases, exposing regions of DNA for protein regulation. So these are post-replication, post-transcriptional processing. Post-translational processing is after the protein has been formed and glycosylation things take place. Yeah, and the hydrogen bonding gives the DNA specificity, but what gives the DNA molecule stability is the base stacking and the hydrophobic interactions between the different bases. Okay, when they're stacked stacked up, and that's quite important. So bases are stacked parallel, allowing hydrophobic interaction between them. So this gives the DNA its stability. And there are about 10 bases per turn. Okay. Now it varies, I think, between 10.1 and 10.6. So about 10.4 is the average. And the chains run in opposite direction. I think this is space group C2. So the chains were in opposite direction. One goes from 5 to 3, the other one goes from 3 to 5. And yes, the B helix, which is the main form of DNA, is a right-handed helix, and a consequence of the double helical formation is that you get these major grooves and minor grooves, and these grooves allow penetration of regulatory proteins into the DNA, and you can see the major groove facilitates that better. But the bDNA is not the only form of DNA. There are these other forms as well. There's aDNA, which is a much fatter type of helical structure. It's related to helix formation in RNA because of the extra hydroxyl group on the ribose that tends to give fatter helical formation, and that seems to relate to this aDNA form. There's also zDNA, which occurs particularly in plasmids, which is very different. It's a left-handed helix, and this is a table taken from Wikipedia, actually, which compares the different forms of the DNA. This is the main structural form of DNA, with the bases inclined at a small angle to the horizontal, so 1.2 degrees, whereas the A form and the Z forms, the A form particularly, has a much greater inclination to the main axis of the helix. And zDNA as well, that's got an inclination of -9 degrees. But bDNA is the main form. As I said, zDNA you find in plasmids, and you can get switching between the forms depending on the degree of super twisting, and we'll cover that in a minute. And also zDNA can form in response to high ionic strengths, and that affects the phosphates, okay, the sugar phosphates. So these different DNAs, zDNA and aDNA, can be important in terms of regulation for gene expression. Right, now another interesting consequence with DNA. Well, first of all, RNA can form these hydrogen bond structures within the same chain. RNA is a single chain molecule, but you can still get hydrogen bond structures. If you've got these complementary or inverted repeat sequences of the bases, these are called palindromic sequences. So you take a point in the DNA or RNA molecule. And let's take the mRNA here, and you read in opposite directions. Then you see you've got a C and a G, a U and an a G and a C, an A. Where are we? 1, 2, 3, 4. 1, 2, 3. A and a U. So you've got potentially base pair forming nucleotides on different parts of the chain, different local parts of the chain. And if the energy conditions are right, then you can get this hairpin form, of course leaving unpaired bases at the end, because physically you can't base pair all the bases. And this occurs also, or can occur also, in a DNA duplex, double chain DNA. So here we've got this inverted repeat sequence. Actually, the inverted repeat comes from this. If you go G, T, C, A, G, then on the complementary chain here, it goes G, T, C, A, G. And here you've got G, C, T, A, don't click, C, G. So if the conditions are right, then this inverted repeat can lead to the formation of two hairpin loops, one in this chain and one in the lower chain, and we call this a cross or cruciform type of structure. So it's possible, if the energy conditions are right, then you can get this cruciform structure. And this relates with the super twisting, the compaction of the DNA molecule. And the significance of this is that if you get cruciform structure formation in DNA, you expose regions of single strand unpaired base DNA within the DNA structure. And that can be important when it comes to regulatory proteins which recognize single-stranded DNA. So cruciform formation can be an important part of gene control and regulation. So as a should I tell you or not? As a gentle hint, you should look for these structures, look for these sequences in question 2. Right, that's as far as I can say on that one. Right, so a tRNA, whose structure was worked out in the 1970s, also have these regions of, within the same chain, hydrogen bonding within the same chain to form significant structures like the anticodon loop, which contains these 3 bases, and the acceptor stem, which links onto the relevant amino acid which is being brought in, and also these other loops, the D loop, variable loop, and this T psi C loop here. So it's got these, it's quite a nice structure actually, the RNA, with also some modified bases as well, which enhance the stability of the tRNA structure. And interactions between the different loops arms also enhances the structure, stabilizes the stability in terms of the energetics of the process. I like tRNA. In fact, on the Rasmol, we've given you a tRNA to explore. I think so. Yeah, of those molecules that we gave you. Right, super twisting of the DNA duplex. So this is what happens is you form the double helix, but then you can actually twist the helix. This is the first stage, in fact, in condensing the DNA. This condensation process in eukaryotes is then further enhanced by the addition of proteins like histones. But super twisting is like the first part of the condensation, the compaction of the DNA molecule. And then because of the energetics, after so much super twist energy has been absorbed, it then becomes more difficult or unfavorable to twist the DNA anymore unless things happen. And supercoil formation is one. Cruciform formation can be another, depending on the conditions. You don't use regular phones now. Everyone uses mobiles, but in the days of regular phones, people wander about and all this sort of stuff with the cords, and they get twisted up, and then you get these coils form. And the interesting thing is, if you used to pick the phone up and let the receiver dangle down, and then you see it untwist. That was the energy being released in the phone. Something similar to that. So super twisting is important in terms not only of packaging, but also important as a mechanism for controlling gene expression, because it can be used to stabilize more open conformations such as cruciform formation. All right, and there are these enzymes called topoisomerases. Again, did Kath talk about that for the ABTs? Topoisomerases? Nope. Okay, but these help to control the number of topoisomers, which DNA molecules differing in the numbers of super twists. And they do this by catalyzing the breaking and reforming of the DNA strands. So, and you can see the action of topoisomerases using agarose gel electrophoresis. So when you have the demonstrations of electrophoresis from Jan, which I understand will be quite soon, I think she'll be covering the protein electrophoresis, which we did yesterday, which is SDS gel electrophoresis, with the DNA electrophoresis. In fact, DNA is good because you don't have to treat with any SDS and things, because DNA already has the uniform charge per unit length facility because of all the phosphates on there. But they're generally much longer, bigger molecules, so acrylamide won't do as a separating agent. We use more sort of looser gels, which are generally agarose. Agarose is the usual gel medium for DNA work. There are others as well, but agarose gel electrophoresis. So Jan will be talking about this. So this is an electrophoresis setup using agarose gel electrophoresis, and depending on the numbers of supercoils tend to give a more compact structures. So more compact structures move faster on a gel for the same molecular size. So this shows a fully supercoiled form, and this is a fully relaxed form, depending on the action of the topoisomerase. Yes. What are the girl's best friend? Dimers. That's right. Yes. I'm sure it was something else, not dimers. I can't think what there might be at the moment. But, right. So many DNA binding proteins are dimers simply because, yeah, it's a double helical molecule. And these dimers have subunits which can be non-covalent or covalently attached. This is the non-covalent example here, and this is the subunits showing this this symmetry. This looks like mirror symmetry, and this is important. See how this fits into the major grooves in terms of regulation. So many DNA binding proteins are dimeric, okay, but not histones. They contain these are several subunit structures, and histones are highly cationic for a reason, because they're designed to pack the DNA in eukaryotics, not prokaryotics. Don't find them in bacteria. So I mentioned super twisting as a way of first stage in compacting the DNA. In eukaryotes, histones complete the job, and they form the chromatin structure. So histone, this DNA, is the chromatin structure. And that's why, because they're very positively charged, like yesterday I mentioned, the histones don't work on STS electrophoresis because of the very high positive charge. Through amino acids like lysine and arginine. That's right. Yes. There were one or two unimpressive responses yesterday when we were doing the Perkins demonstration, where I asked people what particular amino acids coming up were. I think we got cysteine out all right because of the sulfurs. Methionine seemed to fool a lot of people, and one or two of the others as well. Just as a warning, when it comes to next September, when we have the final events of the MSc, unless you've got an extended project, then that's different. The sort of questions the external examiner asks, like, you know, amino acids, sugars, all these sort of things, if you know the basics. And there's no formal interview now with the external examiner. What happens is he goes round the poster displays and targets everybody and fires questions which are completely unrelated to the poster, just testing your general sort of knowledge. So it's good that you are on the ball with your amino acids. Just as a polite warning. One or two came a cropper last year by the unexpected question, which you should know anyway. So what I'm going to do is just